Okay, so we're just going to get started now. I'm Laura Occhuzzi. I work with the Life Raft Group. As I said, I'm Italian. I have been raised by, and I've had strong Italian women around me my entire life. And it's probably the third time I've been to Italy. And every time I come here, I just still get chills between the history, the beauty, and the amazing food that I was raised with and learned how to cook. And I refuse to go to eat any place near a monument because it's just not good. You have to go, if you're staying, go walk around into the small little areas and you'll be amazed by the food that you eat. So I'm so glad that we're here. I'm so glad that I'm surrounded by familiar faces. And I think we're going to have a really good session this afternoon. So with that, I'm going to turn you over to what I think is SPAGN royalty, because Kai's been around for a long time. So Kai, when he's ready, he'll begin to introduce the presenters. Thank you.
I had the same water. Here we go. Okay.
I don't know what we would do without tech guys.
Okay. Yeah. Hello from myself also. I'm Kai Pilgermann. I'm also a German GIST patient. And it's a pleasure now to get Eva Wadelmann here for the talk. She talked yesterday already. I think the most people enjoyed that already. And now we'll see how deep she can go into the GIST part after the student lecture yesterday, now the expert lecture, I guess. So welcome. And
yeah, thank you so much. It's a great pleasure to be here. And I think I switch over to the other side so that you can see your audience. I also will talk loud. So I think you hear me all in this small room. Yeah, yesterday we had already the session about the molecular pathology, and it might be that you thought in some minutes, oh my God, this is so complicated. And it's also it is complicated. And I thought for those who were not there, who just quitted that session from the beginning on, I just start with a small part about how we do this molecular analysis of GIST, and afterwards what it means for treatment and recurrence and so on. So please interrupt me once you have questions and don't understand what's going on. I also don't understand why it's not going on here. Do you think that this is already... Of course you can have my slides. Yeah, no problem. Yeah. You can read it night and day and look again and so on. So, oh, that's then ideal. So, now it works. Very nice. Thank you. So we yesterday you heard already. We begin with a paraffin block. So the material where the tumor tissue is in, which is wax, if you want, and you can see that here is the block, and inside this brownish thing in there is the tissue. And also not only for looking under the microscope, but also for the molecular approach, we make slides. It means we make thin slides. For the DNA extraction, it's up to 10 micrometer, which is very, very, very thin. And then we make several slices, and then we scrape off the tissue from the slice. You could say, why don't you just take the slice and put it directly into the tube? The reason is that often we have to do some macro dissection. It means that we take only the tumor tissue to look for, and on such a slice there may be normal and tumor tissue. And if you contaminate the whole probe with your normal tissue, it might be more difficult to find the mutation. So this is why we try to concentrate the tumor tissue, and this is why we do it first on the slides, on the glass slides, and then we make on a H&E stain slide some marks so that the technician knows which part of the sample she should or he should microdissect. Afterwards, we have to get the wax out of the sample. Otherwise, we cannot process further. So we have to deparaffinize. Our wax is called paraffin. So we have to get it out with some chemical approach. Here's the macro dissection. You can see it is scraped off the glass tissue, and then the samples are in the tube. So from that point on, we have to get the DNA out. And again, this is, of course, a chemical procedure with fluids and hours waiting for the next step. And normally today we can do a lot with robots, so robotic technical work, so that no one has to do the whole time with hands-on the procedure. But here you see this is an historical image. We do it still with the hand. And this is also historical sequencing, Sanger sequencing, which has been done for years and years and years. And the reason why we did that is cheap, fast, and easy, but only 1 exon per run for those who were there already. You remember that the genetic information is in small parts on the DNA, and every part of a gene has more than 1 exon, is called exon. This is the coding DNA. And in GIST, we know that we have 21 of these exons in the KIT gene, but only in some of them there are mutations. Obviously, but because if there are changes in the other exons, it has no effect on the function. So it doesn't matter whether there's a mutation in such a non-hotspot region. And here these normally a little bit more colorful hills, each of them is representing 1 base, 1 nucleotide, and altogether there is the code for how the KIT receptor is constituted. And if you change 1 of these, only 1, then it might happen that the KIT receptor is turned on and you cannot turn it down again. And this is then the beginning of a tumor growing. It's not only, in all cases, only 1 nucleotide. It might be also that more nucleotides are missing or are duplicated. So there are multiple types of different variations, and it's still in the research which mutation has which biological impact. Are there more aggressive ones or less aggressive ones? Is the specific mutational type responsive to imatinib, to Gleevec? All these questions have to be answered, of course, and have been answered already. So the modern approach is that we do not do exon per exon because it takes a long time and a lot of hands-on time, but we do the so-called next generation sequencing. And I suppose that you have heard this next generation sequencing several times now, and always you feel a little bit like, ooh, what is it? So I will try to explain it. It's abbreviated NGS, so the experts talk about NGS. And what happens is, again, we have first of all the tumor sample. We have to get the DNA out, but then the workflow is different to what we did in the Sanger sequencing, because in this approach we can analyze all the exons in one run, which makes it much faster. And it's not only 1 gene that we look on such an NGS platform, but several genes. The big panels right now, which are really huge, have more than 600 genes in 1 run. However, these several steps take a time. And so for an NGS result, normally you wait longer than for a Sanger sequencing. And it's much more expensive. So we will come to the costs later on. And I can tell you already, I cannot tell you in euro or dollars or whatever how expensive it is, because it's complicated and it's different in each country. However, if we have this whole preparation of a sample, we put it in such a machine, and this is just an example, not an advertisement for Illumina. There are also other machines doing the same, and we get a complex result. And because it's complex, we now need also biologists and bioinformatics helping us to understand what we get out. Pros and cons. Why should we do next generation? I'm a clear green person, not the red one. But however, we have to see both sides. the good thing about this next generation sequencing is we need only a little amount of DNA. This is helpful if we have only a small biopsy. We can still do the analysis. And before, in older times, often we had to say, oh, we couldn't do it. It's not enough tissue. It's more sensitive, so we find also minor clones, only small proportions of mutations. And in some cases, which we get older samples, especially, which are called wild type GIST, so no mutation in the known regions. When we again now examine them with NGS, we find a mutation. This might explain why some of the patients having a wild type GIST respond to imatinib, because they shouldn't respond. But they respond because in the primary analysis, the mutation was missed due to technical reasons. So if a wild type GIST is coming to me in my institution right now, I first do NGS and look for KIT mutation, because this is normally what then happens. Yeah. Yeah, absolutely. Yeah. Yeah. The problem about the NGS, I will come to that, is it has also some disadvantages, also again leading to wrong or false wrong results. So this is the thing with the wild types. And then what also is good about this issue, of course, you've got other genes. So it might be that there are other mutations synchronously with the KIT mutation, for example, leading to a diverse biological behavior. You can quantify how much mutant DNA you have, and you can also find resistant subclones, which is also important. I come to that at the end again. But the disadvantage is, because this is all in a machine, you cannot see the colorful hills like in the Sanger sequencing. There is a complex bioinformatic information coming out of the machine. And depending on the algorithms you use, which is done in the computer, to find the right sequence, it might happen that long deletions in the KIT gene are missed. What is a deletion? A deletion means that a part of the gene is missing, is lost. And this deletion might lead to an activation of the receptor, which sounds a little bit stupid because something is missing. But sometimes it's just the frame that is missing, as it is the case in KIT. So due to the algorithms, it might happen that you say, oh no mutation in KIT, exon 11. And then if you look with Sanger, then you find the deletion. So what we do, if we have a case, classical case, small bow, 10 centimeters, spindle cell, proliferation is rather high, and I get the result, it's wild type and KIT exon 11. We now do routinely as a reflex testing afterwards a Sanger sequencing to see if really there's no deletion. So longer deletions may be missed. So you do not always have to believe the NGS results. We have more bioinformatics with an NGS. We have higher costs, and the turnaround time is longer. Although we have not exon per exon, only the process of NGS is already time-consuming. What about the costs? I used to have here a number of euros, but I took it out because I know right now it's, yeah. We, as an institution, I would say 2015 or so, we started, but it is a development. We started with 26 genes, and now we are like 638 or so per panel. So it is a development. Also, of course, of the suppliers of the machines, and so they also develop, but we also developed and thought it would be good to have more genes on the panel. So it varies throughout countries what it costs. In Germany, it's especially complicated due to different health insurances, which very different approaches, what they should pay, what they not should pay. Then it depends also in Germany whether you're an in or an outpatient, so whether you're in the hospital or just at your oncologist, practical oncologist. And the larger the panels get, the more expensive it gets, just because the chemicals are more expensive and it takes longer time to analyze and so on. So, yeah. it's a dynamic situation, I would say. Perhaps we can discuss that at the end. What we learned from all the years of working on that, we have now nearly 4,000 GIST in our registry in Münster, is that, as we knew before, KIT exon 11 is by far the most common subtype. 62% of all mutations are found in that region. Then we've got the nearly 9% for exon 9, and we've got a large part of exon 18, which is, if we take that together, like 12 to 13%. And then we've got this group of wild-type GIST, so-called wild-type GIST, 12.4. And the question is, who is in that group? And I will go through the other genes briefly. And what you also see is that the other ones, KIT exon 13, 17, and also 8, are very, very rare. And also the other exons and PDGF receptor alpha 12 and 14 are very rare. So, of course, the more rare the subtype is, the less experience you have with the response to treatment or the aggressiveness or whatever biological behavior is. What we learned from all these tumors is that different mutational subtypes in different parts of the GI tract. For example, in the rectum, which is here on the right side here, you will never, I say never, find a PDGF receptor exon 18 mutation. They do not exist there. So if I get a case with the report it's a rectal GIST and I have an exon 18 mutation, I would say no. It's not possible. And the same holds true also for other subtypes. So what does that mean? Obviously, the different biology in the different parts of the GI tract is in part also influenced by the mutational subtype. You can see the blue, or gray blue is it here in this room. This is KIT exon 11. And you see, okay, this is small bowel is as expected 63% and stomach 60. And in the rectum, it's more than 80. It's 80. It's 80. And also in the esophagus, it's not shown here. It's really a huge number of exon 11 mutation. And then the specific thing about the small bowel is exon 9, shown here, 23%, nearly a quarter, is exon 9. And we know from exon 9, it has implications for biology and also for response to imatinib. It's a different situation. And for the stomach, we've got here this nearly quarter of PDGF receptor alpha, exon 18 mutant GIST, which often can be resistant to imatinib. So we have to know where's the tumor, what is the biology, and interestingly, if we take, for example, exon 9 mutations in the stomach, they are very small proportion. They all are very low aggressively. So obviously, the same mutation doesn't mean the same in a different part of the GI tract. And this is from Heike Yuensu and out of the SSG18 study. And it seems very crowded here, as these curves are often, because we try to show how different the biology is depending on the mutational subtype. And you can see, here up is the PDGF receptor alpha group. They are probably prognostically the best tumors. And the exon 9 cases are down here. And all the rest is in between. So we are always wondering and still discussing whether it would be good to include the mutational subtype in the risk assessment, because it might make a difference. Yes. Normally, we say in the primary situation, without any treatment, you've got one mutation. One. If somebody wants to sell you 2 in KIT, then there's a mistake. So, and also, you do not have KIT and PDGF receptor alpha. Either you have the one or the other. So it's exclusively. A difference is if you are treated. If you are treated, then subclones can develop, but without treatment, they will not develop. So it's just the one mutation you have. No, they're not subclones. The subclones develop due to the inhibition with imatinib, for example, because then there's a selection pressure. It might be that in the primary there are subclones, but they are not superior to the others. They have no reason why they should grow quicker than the others. So probably there are some very, very small amount of double mutated cells already in the primary, but they will never grow up because they have the normal only one mutation carrying tumor cell is superior, is growing quicker. But once you inhibit the single mutation cells, then these double mutant suddenly have an advantage, and then they grow up. This is why probably subclones are there already. We cannot detect them, and they are meaningless because it's just by accident that some cells have two mutations. But if you inhibit the single mutation, then the doubles come up. It's one theory, at least. So what about morphology? I'm a pathologist, so I look on the microscope and I have an opinion on a tumor, what I have to expect which biology, which mutation. And as we are all talking now about AI, we talk about the possibility we scan that slide and the computer says, oh, this is a KIT exon 11 mutant GIST. Let's see whether this happens in the next 10 years. But we knew already 20 years ago that obviously there are different different morphologies. There is the spindle cell type and the epithelioid one. And this was also the beginning of my interest for GIST. This is why I show this, because at that time we didn't even know that there are PDGF receptor alpha mutations. They weren't described at that time. We just saw we had tumors with KIT mutations and others without. And the KIT mutant GIST were spindle cell and the others epithelioid. So obviously there were two different types. And then shortly after that, PDGF receptor alpha was detected as a second important gene. And again, we could see they have a different morphology. They have more rounded tumor cells, not spindle, and they have giant cells, which we do not see in KIT mutant GIST. So obviously we've got both tumors being dependent on the KIT receptor, but different mutations lead to different phenotypes. And of course, this might help one day to do it with AI without a pathologist sitting hours and days on such a sample. Unfortunately, the quality of this beamer is but however, what you can see, the pictures, these 6 pictures look different. And all are GIST, they have different mutations, different locations. So the morphology is very diverse. And especially for the epithelioid ones, for people who do not often see GIST, it might be very difficult, if even impossible, to recognize this is a GIST because it looks so bizarre. It reminds you of a neuroendocrine tumor, carcinoma, whatever, but not of a GIST. So we use, of course, established markers I will not mention here. What we learned from that is morphology correlates with the mutation. So from the morphology alone, you can already suspect that this or that mutation could be there. Then I talked about this wild type group. It depends always a little bit on how many cases you looked for, how huge this group is. Something between 10 and 13%, I would say. And of course, these are those where we do not know what to do with them. So it's important to understand that group. And I will just quickly run through the genes that are instead mutant and obviously can also lead to a GIST. One is neurofibromatosis type 1, and these mutations are rare, of course, much more rare than those in the other genes, but alternatively can be a reason why people get a GIST and even more than one GIST. So with NF1, also multiple GIST can occur during lifetime. There are some papers, as this is rare, it's difficult to calculate, saying that the frequency of GIST in NF1 patients is 7%. For the single patient, this number is meaningless because am I with a 7 or am I with a 93? So, but it is just, the message is, if someone has an N of 1, then the risk is increased. So it should be looked for, whether it just is developing. And interestingly, these just are normally in the small bowel, not in the stomach. The other gene is KRAS, and we know KRAS from other tumors. Here is a small series, but as you can see here, these are the KRAS mutations, and these are KIT or PDGF receptor alpha mutations. Only one case has no KIT or PDGF receptor alpha mutation. So I would say this is just a bystander mutation. The main mutation is this one here. So primary KRAS leading to a GIST, difficult, very rare. This is in difference to BRAF. BRAF is another gene. I know that all these names are disturbing, but we finish that soon. BRAF is also in the same signaling pathway as KRAS and NF1. So the tumors that develop are very similar, and also these BRAF mutant GIST are all in the small bowel. Here's one example of the stomach, but normally they are in the small bowel, and they do not have KIT mutations. They do not have PDGF receptor alpha mutations because they have BRAF mutation. And there are BRAF inhibitors and also other drugs now which can lead to response. But again, it's very, very rare. If you look at the number of our own series, 179 wild type GIST, which is already a huge number because they're so rare. And among them, 4% of BRAF. So very small group. However, with this multi-gene analysis we do now, it's no problem to look at synchronously whether such a mutation is there. And then another issue is this SDH. SDH is always mentioned as another important group of the non-KIT, non-PDGF receptor alpha GIST. This here is a mitochondrium. Those of you who remember biology, remember that in the cell there are these organelles, having they are responsible for energy, and the Krebs cycle or citric acid cycle is the way how these mitochondria produce energy for each cell in the body. And they need enzymes for that. And these enzymes, one of them is the succinate dehydrogenase, which is, of course, an awful name, and I need, I think, a little bit of water if you have, otherwise, yeah, I take the bottle. Sorry for that. So SDH is an abbreviation. It's much easier to say. And SDH is so in the cell. And if there's a defect in this SDH enzyme, then there are several molecular events in the cell and simulating hypoxia. And by any way, this might lead to the development of a tumor. And SDA is the abbreviation for this complex. And there are several partners in that complex, A, B, C, D, 4. And SDHB is one of them, and we can stain for this protein by immunohistochemistry. So the normal body cell has SDHB, and the deficient GIST has a loss of SDHB. So this whole complex is not working anymore, and this is one reason why GIST can develop. It sounds a little bit bizarre, but it's like this. So a group of Germany, in fact, Florian Haller, has detected that this must not always be a germline mutation leading to this, but it might be also another dysregulation process leading to such a loss and then leading to GIST. How often do we have these alterations? It's always a matter of how you count cases. If we look at these cases here, from Inga Marie Schäfer, some of you might know her, then we've got in this series KIT mutations in 75%, PDGF receptor alpha 10%, and then in this series they have even 13% of SDH deficient GIST. And a very interesting but complicated scheme is here, because here is shown what the specific things about these tumors we know. For example, they often occur not only one nodule, several nodules. They occur nearly exclusively in the stomach. Young female patients. They often develop metastasis. And although they have metastasis, the the biology is rather indolent. So with these metastases, they can live years and years and years and years. And the bad thing about these mutations is the tumors are not responsive to imatinib. So yeah, two sides of a medal, I would say. And if we compare these specific things about these tumors with all other GIST, then it's obviously a separate group which needs a different treatment, a different survey, probably a different approach. And about genome, yeah, of course.
I have diagnosed as fine with a GIST. And there, at the moment, I have diagnosed there, it was either in exon 9 or exon 11. Everything else was a wild type.
Yeah.
Nowadays, we have different GIST mutations that we can analyze and name, and also the treatment of all these different mutations is different. What exactly is then the reason why all these different types are named as a GIST? They are, as I understand, completely different mechanisms, how they start and they grow.
Mm-hmm.
They all the same disease GIST.
Yeah.
And all the same disease.
Yeah, you're completely right. This is somehow disturbing because you think, why shouldn't we separate that and give them different names? The common thing all these tumors have together, that they have specific proteins they express, and this is why we lump them together. And the most important one is DOC1, indeed. It's a protein sitting in all these tumors, independently of whether they have KIT, PDGF receptor alpha, NF1 or BRAF mutation. So they look very similar, although I showed you pictures where they look so different. But altogether, they are all mesenchymal tumors. They occur exclusively in the GI tract. They express mostly the KIT receptor, and even more the DOC1, which is the abbreviation for detected on GIST1 on a cell line, and turned out to be on all these GIST, independently of what they have as mutation. And this is the reason to group them together. But you're right, from the grouping together, we have to separate them again and to say this is type 1, type 2, type 3, and so on. And the more genetics we perform, the more subtypes we create, not only in GIST. It's also true even for colorectal cancer, where you would say, well, this is a homogeneous group, isn't it? It is not. It's so diverse that although they may be lookalikes on the genetic level, they are completely different. It's difficult to understand, but the issue is that, of course, it's also historical development. We started with exon 11 and then 9. And then more and more of the gene would be analyzed, and so also the rare types came up. But the signaling pathways also downstream of these activated different KIT mutant GIST is the same. So what happens after the mutation is very comparable. This is another reason to lump them together. But you're right, it's a very heterogeneous group, although it sounds as it would be one tumor. What you can see here is that a lot of people thought, okay, as KIT and PDGF receptor alpha mutations occur so early, even in these small microgist, as an initiating event probably, how is the tumor progressing? And why are some tumors metastasizing and others don't? And again, there has been some work, not only from Inga-Marie, but also from other groups, showing that there are additional aberrations on the genomic level leading the progression. And here I mentioned only some of the tumor suppressors which are lost, for example, MAX or DMD. These are proteins which are lost during the progression and making the tumor more aggressive. So we know this already, but it's very difficult to address these by treatment. And nowadays, this is from last year, life is getting even more complicated. We heard already that we do not make panels anymore. We make genomes, exomes, and we do methylation arrays. And This is just a very colorful picture. We cannot go through that. But what they did here is they compared a specific subgroup of exon 11 mutant GIST with 557, 558 deletions with other GISTs and found a lot of differences, not only different mutation and KIT, but also in other chromosomes, losses of parts of chromosomes, exchanged nucleotides, different methylation. So the whole tumor is different. And the problem is we come from a time where we looked at one gene. And now we learn we have to see at least 20 genes to really understand. And I think next year, probably we need at least 100 genes to understand, and also the regulation of these 100 genes. So it's getting complicated and more complicated to see why biology in one patient is like this and in the other is different. And this is what research has to provide. More information, especially those which are not only describing these complicated situations, but also leading then to a different treatment. And to say, okay, this patient gets imatinib and the other doesn't, for example. That would be one simple example. So if we look at the molecular classification, I'm over the time already, then you can see we can dissect GIST in two parts. Those having SDH, this protein from the mitochondria, or those having it not. If we take those having SDH, then they have either KIT or PDGF receptor alpha mutations, the vast majority, very rare other mutations, BRAF, NRAS, KRAS, HRAS, HRAS, NF1, other very, very exotic genes, and also translocations. We learned yesterday what it is, which are also very rare. And I'm not sure whether we should call these tumors GIST. And on the right side of the picture, you see those having a loss of this SDH, and they can have either germline mutations, so that it's genetic hereditary disease, or they can have a dysregulation of this protein, of this complex, and they have different tumors in the stomach, multinodular, young female patients with metastatic disease, also in the lymph nodes. And I will skip this one because it's too complicated. I would just want to come to this last point. Although resistance is always an issue difficult to discuss. But what I think is very important is also if the resistance occurs, and it occurs in some patients, that imatinib doesn't work anymore, it's important to understand. Otherwise, if we don't understand how this works, we cannot do anything against it. So there are several works from Sebastian Bauer's group showing that if we dissect how the resistance works, then we can also do something against it, and we can choose new drugs. And there are now a lot of drugs, more than 10 years ago, with avapritinib and repritinib and regorafenib, which have changed the situation, the landscape, but it's not my issue as pathologist to talk about that. But it's important to look for what happens. And there is here, for example, this study for repritinib and sunitinib showing no difference between the two groups. But this is because it was not looked for which type of resistance was it. And after dissecting that and looking which subtype of resistance it is, suddenly there is a difference. So there are still patients having a benefit from repritinib. I think you can explain it later. And it's not that the study is negative, as shown here, where no difference occurs. And so there will be a new study on that, together with Essen and Münster, for example, where we look for that, and we want to do liquid biopsy and look whether we can also in the blood already detect these resistance mutations. And I hope, of course, that a lot of patients will agree to take part in that study, because it's very important to understand. If you understand, you can cure. If you do not understand, you cannot help. So my last picture, I think, or nearly last picture, the role of liquid biopsy. We all hope early diagnosis. One day you just take some blood and then every disease is recognized. I think it's too optimistic, but we are on the way. We can see, of course, whether there is any heterogeneity in the tumors if we take the blood, because we think that all the different clones should be in the blood, which has to be proven. We can measure mutational levels and see whether they rise or go down, so whether there's response or not. However, it's still under development. It's not established. It's not daily use. So it's not a routine procedure. We now stick to these genes, and I think these are the most important ones, which we can now analyze rather easily. And this is, yesterday I showed Münster by night. This is by light, and you should come and visit us. Thank you. Questions?
Don't know what we don't understand it.
Yeah, exactly. That's it. But we have to look for it so that we get better. True. All others are already. No?
We're not sure if that's the gist.
And which ones?
A few slides back.
Yeah. Yeah. In that scheme where the translocations. Yeah, because this is what exactly is the question that you posed. And why is it still called GIST, although it does not have the mutations, the right mutations, and so on. And once you then have tumors among them, even not expressing DOC1, of course, you have to argue and say, oh, should we really call it a GIST? What is the argument? So I think some of these weird translocations will fall into another sarcoma group once you look more deeper. Because, for example, there was one with an NTRK translocation. I would never call it a GIST. I would say it's an NTRK sarcoma. Yeah. Exactly. And indeed, when I came to that research on GIST, it was a time where nobody knew what these tumors were in the GI tract, being not a leiomyoma, being not a schwannoma, being something else. And this was the beginning of recognizing there is something specific, and they are much more frequent now than the others, leiomyomas, schwannomas, and so on.
Or whether it's a critical illness.
Absolutely. Yeah. It's very important. And it's important to be sure that the diagnostic criteria we use are really correct. So we always have to ask again, is it really a GIST? That's true.
One more, and then after that we can go. Is there an chemotherapy or else?
Peter's saying no. I think he should answer that later on as oncologist, because treatment is not I wouldn't say it's not my passion, but I do not know enough about it. But Marcus had something?
Yes, they have a brief conversation about the microenvironment. It's becoming more interested research. Do we know anything in GIST now?
There have been some works on that from France, for example, where they looked for tumor microenvironment and whether immunotherapy could be of help. Until now, I would say there is a tumor microenvironment as in any tumor. And my personal view, which is very subjective, there are GIST with a lot of lymphocytes inside. I don't know why, but there are there. But there is no current application of checkpoint inhibition, for example, and all that what we hear from other tumors. But I'm sure if we look deeper, then we will identify again a new subgroup of inflammatory GIST or so, where it could be of help, but it's not yet detected. Okay, so